Welcome, everyone. Thank you so much for joining our webinar today. My name is Susan Chung. I am the Senior Global Product Manager for Quantabio with focus on NGS. So I wanted to introduce to you our speaker for today, Dr. Philip Lavretsky. And he is currently the Associate Professor at University of Texas at El Paso for the past 8 years, since 2016. His passion for evolutionary genetics for mallard ducks started some time ago, and he received his PhD from Wright State University in 2014 with his dissertation that focused on the population and evolutionary genetics of mallard-like ducks. He will be speaking on his exciting research today, which spans across evolutionary, population, and conservation genomic disciplines, where he uses molecular data to ultimately better the conservation practices.
All right. Well, thank you. And thanks everyone that's online right now to listen to me ramble for a little while about ducks and conservation and genomics. The title of the talk is Wildfowl Genomics in the 21st Century. I'm going to focus on a particular species, the mallard, as we've been able to really dive deep into a lot of things that are unseen and unknown with it by using genomic methods. My lab here at the University of Texas, El Paso, we sort of span field to gene. That's our moniker. We do everything from catching all the species that we're interested in to bringing them into the lab and learning about them with molecular methods, looking at their associating that with phenotype, genotype. But then we've also got birds out there with telemetry units, and again, associating genotypes with migration capacity, habitat associations, and so forth. And then we do lots of outreach to really make sure that the community understands what's happening here, and then also engage them in concepts of evolution, conservation, and so forth. And really the question that my lab answers is, what are we working with? Whether it's state and federal partners or NSF or anybody else, what we want to understand is this thing what we really think it is? And to do that, we do this with genetic methods, right? To be able to really understand at fine scale population structure, why species are present in certain locations, so their genetic uniqueness, and how also that genetic variation affects historical and contemporary hybridization, demographics, and so forth. To do that, my lab employs many different methods. We go from single gene PCR and Sanger sequencing when we're focused on a particular locus like mitochondrial DNA or something like that, to partial genome sequencing. And we use pseudo-randomly targeted dd-rad-seq loci, as you'll see in this talk, to get about a half to 1% of all of the genome of the species that we're interested in. But that still gets us 500,000 to 1 million base pairs, tens of thousands of markers, SNPs, to be specific, to be able to do our ancestry calls. You know, we don't need full genomes to do ancestry calls. So you'll see that we've been able to tailor these methods so that way we get thousands of markers for thousands of samples all at once. We also use targeted approaches for sequence capture on some historical data that I'll be talking about. And then, of course, we use full genome sequences. Once we know who we have in our data set, we target that with both Illumina, Oxford Nanopore, and Dovetail Genomics, or Cantata Bio these days, to be able to get at the question of more fine scale resolution of what's occurring at the genomic level. But we focus those specimens once we know what we have in our larger data set using that partial genome sequencing. And a big question in my lab that we study is hybridization. Hybridization and gene flow specifically is thought to have been not really generally important for evolutionary purposes, but now with genomic data, with more and more genomic data, we really know that, in fact, gene flow is a natural process, naturally occurring across all forms of life, including us humans, with Denisovian and Neanderthal genomes admixing with Homo sapien to really that explains today's human lineages. You know, our genomics is really an admixture of these various hominids. In case you're not familiar with the concepts of hybridization and gene flow, I always like to put this slide here so that way everybody kind of catches up. What hybridization is really parent one with parent two. They interbreed and they make this first generation hybrid, or an F1. This, in biological sense, is lost breeding potential. Both species picked the wrong mate. They made an F1 that potentially can't interbreed. And so the male and the female of that generation that picked wrong essentially did not contribute to the next generation's genetic gene pool or gene pool. And so they are essentially what's known biologically as lost breeding potential. What gene flow is hybridization, but then what happens is that first generation hybrid, which is essentially a vessel of genes of the two parents, interbreeds with one or both of the parentals, and in essence, moving the genetic material of parent one to parent two. And this is what is thought to be a conservation concern when hybridization or gene flow specifically is occurring within a system. And the consequences of introgressive hybridization, which is another term for gene flow, is several different pathways that could occur. In one sense, these hybrids could actually evolve into something new, a new taxa. And one of those birds in my systems is known as the Hawaiian duck. It evolved through this combination of two parental species that eventually gave rise to what we consider today as the Hawaiian duck via hybrid speciation. Then you could have things like hybrid swarms or heterosis, where you got hybrids actually potentially doing a little bit better than the species found on the landscape. And we have a system in New Zealand where the introduction of mallards into the New Zealand landscape resulted in a widespread hybridization with endemic gray ducks. But these hybrids are actually showing higher fitness values than gray ducks alone. And then, of course, what we all are largely concerned about is outbreeding depression. When you intermix two distinct gene pools that are adapted to different ecologies or general biology, you essentially could have what is known as genomic extinction, as well as the loss of one or both species. And in this case, we're going to dive into this more specifically with the wild mallard and worldwide mallard populations. If you haven't gotten it to yet, I work with, well, to be fair, my lab works on anything that's got DNA and money, but our main focus is waterfowl, and more specifically, this group of birds known as the Mallard Complex. Hopefully everybody knows the mallard at the top of the green head, who has a whole Arctic distribution, nearly worldwide distribution since we put them everywhere. But essentially every continent and island system even has their own mallard-like duck. This is what's known as a successful adaptive radiation. There's 14 of them. On mainland North America, we have a Mexican duck, two types of mottle ducks, American black duck, and of course the mallard. In Hawaii, we have the Hawaiian duck, and on Laysan Island, the Laysan duck. And so all of these species are uniquely adapted to these various ecologies. And this is something that we study in my lab. More recently, though, I can't talk about the wild complex without talking about the domestic mallard complex. If anybody doesn't know this, the various domestic forms outside of the Muscovy duck, which is South American domestic variant, the mallard is essentially the wolf of all the various domestic birds that you see out there. They were first domestic, and they were one of the last successful domestication events attempted initially by the Ming Dynasty in 500 BC. But since there's just as many domestic breeds as there are of dogs currently. And I'm sure everybody knows what the Pekin duck is. Maybe some of you have eaten them. But then today we're going to be talking about this thing called the game farm mallard. I know it looks like a wild mallard, but essentially this thing was propagated in about the 1650s initially by King Charles II for hunting purposes. Essentially, this breed, instead of being artificially selected for egg production, meat production, or a combination or fanciness, this breed was starting to be picked for flightiness or hunting capacity, these types of traits sit for the last 400 years. Of course, ducks, waterfowl, and humans have been interacting for eons. There's good data that even our Neanderthal ancestors hunted, ate them. The first record of consumption of by humans in Sweden dates back to about 11,000 years ago. Of course, the domestication of them basically interlinked humans and ducks for until today, since then. And they continue to be this interactive model, both agriculturally important, socially important, and economically important. And they essentially also are an important conservation beacon. They were essentially at near extinction in the early 1900s, and through important conservation work, they have rebounded massively here in North America, and they are sort of a beacon of what people can do when they put their minds together. The method that I use for the population genetics work is called dd-RADseq, double digest restriction associated DNA sequencing. And the method was first formalized by DaCosta and Sorensen in 2014, as partnered with us. And essentially, this is a sort of a snapshot. The figure that you see here is the dark lines are where those loci are in every chromosome. It starts at the Z chromosome all the way in the left, and then it goes chromosomes 1 through 29 as you go to the right. And you can see we get a nice amount of loci across all the different chromosomes, and the number of loci that we get are proportional to the size of those chromosomes. In general, you're looking at 3,000 to 5,000 markers, depending on how diverged the groups are, but you're still looking at 10 to 50,000 SNPs. So that way you get really good high resolution when you're calling ancestry. And I want to note my PhD student, as part of a side project, Hernandez et al. In 2021, we kind of had to formalize a better way to do our size selection. And so in that paper, it was with actually mourning doves or the dove complex. I figured out that at the time we were using other types of beads, but we eventually switched to Quantabio Sparky Pure Mag beads, and they work fantastic. We do a 0.8x cleanup. That is as low as I can go with human hands, and without, so that way we don't get that kind of error. And that's been working really great. We've done thousands of samples with this kind of cleanup. We used to do it as gel cuts, but since we moved to this, we took a two and a half day process, and it is now about a 2-hour process for 250 samples. So really great way that we were able to move forward, so we can open up how many samples we can realistically do without a robot in any given week. Right, so these domestic mallards, the game farm mallard, has been introduced basically worldwide. And everywhere they've been introduced, my team has found hybrids. So these are going to be some PCAs based on thousands of markers, hundreds of samples. Here in North America, we'll really dive into this a little bit more. But initially, this is what we were seeing. We had wild mallards, and then we had these game farm mallards. And you see those dots that are in intermediate PCA space are what would be considered hybrids. We see the same thing in Hawaii, where you have Hawaiian duck and you have these domestic mallards, you have hybrids in between. And again, the introduction of game farm mallards into New Zealand resulted in these intermediate forms, or lots and lots of hybrids between what we consider now a New Zealand mallard and the endemic New Zealand gray duck. So let's focus now in North America on the mallard. I could tell you lots of different stories on American black ducks, mallard ducks, or Mexican ducks, but I think this one will really showcase what we're learning, the types of techniques that we're employing to learn those valuable lessons, and where we're going into the future. If you don't know what happened in North America, essentially mallards were not really found east of the Mississippi River, somewhere in the middle there, where you see some of the states highlighted. So east of that, mallards weren't really present natively. They were more vagrant. And so what happened was in the early 1900s, from 1920 to 1960, private, public, and government officials started to pump out mallards, these game farm mallards, about 500,000 a year for those 50 years. At the same time, however, the boreal forests of the Northeast and the Great Lakes region started to get cut down. And that kind of opened up that habitat. And so there was this sort of natural progression of a prairie breeding duck, which is the mallard, to start moving from west to east. So you sort of had these two things that are occurring in this landscape that started to move birds that weren't really on the landscape into there in mass numbers. And some of the work that we were able to publish in 2020 was that while we always heard about these stories, we didn't know if there was a consequence to the release of these birds, right? And so what you see here is assignment probabilities. You're going to see lots of these. I'm sure many of you are familiar with these. However, if you're not, essentially it's posterior probability of assignment to a particular genetic cluster. Here you have 3 genetic clusters: gray, black, and dark gray. Khaki Campbell's is a type of park duck in dark gray. Black would be your game farm mallard genetic ancestry, and light gray would be your North American wild mallard ancestry. And what you can see as you go from west to east in North America, you start to pick up this black game farm mallard ancestry in many of our wild birds. I did forget to mention that if you are multiple colors, like in the Atlantic flyway on the right, you are considered a hybrid. We also partnered with the Smithsonian for the same paper, looking at birds pre these releases. And indeed, what we found was that we were looking at a two-system situation. The American black duck is a sister species of the mallard, and we thought they're going through genetic demise through hybridization with mallards. But in fact, it was the wild mallard that was going through this sort of demise, and not the black duck. And what we showcase is that Western mallards and black ducks today are essentially genetically similar to those of 150 years ago. However, those in the Atlantic flyway have been genetically changed, that those Eastern mallards no longer are genetically similar to North American mallards. And I will note again, this data was based on sequence capture of the same DD-Rad-seq loci that we identified in previous methods, but created baits to be able to capture that from those specimens that we were able to get from the Smithsonian that, again, dated from 1860 and 1910. So before we really got to the question of, all right, what is happening across the landscape, across the world? We know these birds have been put on the landscape everywhere. We really needed a wild genome to ask this question. And that's because many of the genomes, until we published our recent one, are of domestic origin. We were starting to see that there are issues in some of those domestic genomes. And so we put together a wild North American mallard genome. We knew this bird. We vetted it with DD-Rat, our population genetic work, know that it's 100% pure wild mallard. And so we partnered with Quantabio to develop a 70x coverage genome, really great boost, over 94% BOOSCO score. I'm not going to get into the details of this, but I hope this serves as a resource for anybody that's listening out there that is maybe working with waterfowl. Hopefully you'll be able to use this work. And as I mentioned, we started to see issues in some of the results that we were getting from some of the domestic mallards. And these are PSMC, so demographic analyses, so looking at effective population size, or the size of the breeding population through time. On the x-axis is time, on the y-axis is effective population size. And the big thing that you should know is that all of your domestic birds, which are laying duck, Pekin duck, the CAU wild duck, which was actually made of unknown origin, show distinctly different demographics, right? So you would conclude that maybe mallards went through a severe bottleneck nearly 1 million years ago. And in fact, our mallard shows an exponential increase in their ancestry, with today's effective population size being 4 million rather than 0, essentially. So again, it's important to note that domestication really does cause issues in your genome, that you could have biased results in things like demographic analyses or evolutionary questions. And I'll get to that more in the further slides. All right, so what are the genetic consequences from introducing domestic game farm mallards worldwide? We have our genome that allows us to align all our partial genome data sets together. So let's go out and get a whole bunch of samples all over the world to be able to ask this question. In our communications biology paper that came out in 2023, we got about 1,916 samples, known game farm mallards, known feral mallards from the wild across Eurasia, as well as across North America. Of course, heavy on North America because that's where we sampled the most. But our partners throughout Europe were able to get us nice sampling across their regions as well. We use the same dd-rad-seq method for this analysis. This was comprised of birds sampled from anywhere from 2000s to 2020. We essentially have sequences from every Illumina machine you could imagine, from your HiSeq, 2000s or 1500s, 2000s, 2500, 4000s, HiSeq X, NovoSeq X, and so forth and so on. All of that data is compiled all together, and it all works with our method quite well. And so we were getting really great data, about 340,000 base pairs across those autosomal loci, lots of Z-sex chromosome loci, and also W-sex chromosome link loci were recovered here. Population structure that you'll see is based on about 637 independent biallelic autosomal SNPs. Some of the analyses that you're about to see on the population evolutionary analyses that we were able to do, we did Sanger sequence of mitochondrial DNA. You'll see a network analysis of about 600 base pairs of a control region using PopArt. Again, about 3,600 biallelic autosomal SNPs. You'll see some PCA analyses, admixture analyses, but also evolutionary analyses. We use the entire autosomal data set of 340,000 base pairs to do some tree mix analyses, PSMC-like demographic analyses, as we just talked about, and you'll see a bit about on this gradient forest or